All right, so the experiment that we're looking at is cleavage of structural proteins during the assembly of the head of the bacteriophage T4, and all this work was done by UK Lamelli. So his objective of the experiment was to determine how his four major proteins affected the growth of the bacteriophage head, and obviously all four of these proteins for this were structural proteins. And so what he did, some background information anyways, is that he had identified many different genes, obviously, that were involved in this. All of them play major to minor roles. And so obviously all those ones listed play a role, but the major ones that he had identified are genes 22, 23, and 24. And obviously they encode for structural proteins. And there's one other protein called IP, but at the time of his experiment, it had an unknown genetic origin, but that didn't actually affect his experiment. But the reason why these four proteins were so important is because he had discovered that right before the assembly process would actually begin, that all the proteins would be cleaved in some way, shape, or form to help essentially build the head. He didn't quite know how, but he at least identified that happened. And any protein in this will be designated as P22, 23, or 24 are examples, but a protein will be P and then whatever gene number encodes for that. So like P22 is a protein from gene 22, et cetera, et cetera, et cetera. So how did he do this? Well, he had four different methods that he ended up using. So the first one was an improved version of gel electrophoresis. And what this did is identified all of the different structural proteins that were involved in building up the head. And how this worked exactly is he combined gel electrophoresis with sodium didecyl sulfite, or just SDS, and it allowed the SDS to break down the proteins into their peptides, and then this allowed the pieces to move according to their molecular weights, as how gel electrophoresis normally works if it's not being based off of charge. And all of these steps, by the way, all have a gel associated with it, which when I get to the gels, I'll explain a bit more of how everything worked. So the next thing he did is he had to identify which proteins came from which genes. And the way he did that is he would compare the gel of a wild type bacteriophage to mutants of whatever genes he thought might have had a problem. And these mutations were called amber mutations, and that's because an amber mutation mutates a gene in a way that it can still at least produce part of the protein, which means that in the gel, obviously, it's not going to move the same way as the full functioning protein would, but it's a note that at least it produces a part of the protein. It's not like it makes the gene inactive at all. And the next thing he did is that he looked at how quickly did the cleavage of P22, P23, P24, and IP were. And he used a pulse chase experiment to do that. So he obviously had pulsed all of the proteins before the cleavage had started, and then he timed from that point until, roughly at least the end of when it was to the end of it, so he at least knew how long it took. And then another interesting little thing that he did, they didn't necessarily have to do, but he also determined that it appeared that, from what I could tell, that before the cleavage had actually happened, that all of the genes were combined into a much larger structure, which is another interesting fact. And he ended up doing that by treating it with SDS in room temperature. And he compared that to samples boiled in SDS, and then obviously to just mutants that he wanted to examine. So the first two gels we have is the structural gel and the genetic gel. So the structural gel, what you want to pay attention to here is that, first of all, in lane A, that's the wild type. That shows just all of the proteins from everything. And then as you go down, the one you really want to pay attention to is B, because what B is showing is B shows just the proteins from the head. And obviously C, D, E, and F all show different proteins from different parts, obviously, but it goes to show how well his method worked, because as you can see, there's no X1 or X2 band in lane B. So it shows that he really did do a good job at purifying all the different proteins. And you can see that there's an IP star at the bottom there, which we will get to in a little bit. And then we have the genetic gel, and so in the genetic gel, what you're seeing here is the plus at the top corresponds to a wild-type bacteriophage, whereas 20, 21, and all the other numbers correspond to a mutant that is deprived of that gene or has the amber mutation that I spoke about earlier. And as you can see by looking at the gel, this allowed him to identify which protein corresponded to which gene, which ultimately allowed him to also determine, which proteins are the most important ones, obviously. And in the genetic gel image specifically, now you can start to see P23 star, P24 star, and IP star. And the important thing about what those are is those are just simply the cleaved proteins that came from P23 and P24, which also came from the genes prior of gene 22 and gene 23. So don't be too confused on that. But one thing you also might notice here is that P22, there's no cleaved version of it. And that's because he was never able to actually detect P22 cleaved. He was never able to. So he just theorized that as soon as it's cleaved, it's incorporated into different parts of the head, which is why he was never actually fully able to identify a cleaved version of P22. And so the next two gels we have are the cleavage gel and the oligomeric gel. So the cleavage gel is attempting to show the different times between when you had the first protein made and then when its cleaved version appeared. And at the bottom shows times, so 15 minutes, 16 minutes, 17 minutes, so on, all up until 30 minutes. And he has all of them, all of the different bands that correspond to the different proteins labeled. And just like in the genetic gel, there is no P22 star, and that's because, as I mentioned earlier, he was never actually able to fully detect a P22 cleaved, so he couldn't, obviously doesn't appear on the gel, and that corresponds and kind of backs up from the cleavage that he couldn't really find anything. And also, another interesting thing to note here, though, is that when he was running these cleavages, he discovered that the rate of disappearance of P22 was actually relatively the same as P23, which is interesting. He didn't quite know why that was exactly, but it was another interesting fact that he noticed. And now, moving on to the oligomeric gel. So, in this gel, this might look a little confusing. So, in lane A, at the bottom right, that shows essentially the mutants or the phages being held in and being treated with SDS at room temperature. And the reason why for that is because these proteins normally would exist by themselves in a monomeric version independently of each other in SDS at room temperature. And B, on the other hand, was supposed to, and what B has is B shows all of the proteins together being treated with SDS, except they were boiled in SDS. And you can see there's obviously a huge difference in numbers of bands that are shown in the oligomeric gel. And you can see that he only has P23 and P22 labeled. And again, those numbers on the bottom correspond to times, so 17 minutes all the way up to 30 minutes. And just overall, this is just showing that it seems that all of the proteins are together before the cleavage happens, because otherwise, if they were just by themselves, you would see bands corresponding to the P23 star and maybe even the P24 star. You would see those bands, but he did not see. As you can see, those bands do not exist; they're not there, which can be concluded. One of two things can be concluded from that: either that all of the proteins are together in an SDS-resistant form before the cleavage happens, or after they're cleaved, they're immediately turned into an SDS-resistant form, which is why the bands didn't show. Now, one thing you might be wondering about is, well, what about the smaller fragments? Because if proteins are being cleaved, you're going to have two parts. Well, that was something that he didn't understand either, because the proteins, he obviously had the bigger versions, which are the ones shown in the gels, but no matter what he used, no matter what percentage of polyacrylic gels he used, he could never quite find these smaller pieces. And his best guess, or educated guess as to why it happened, is that the smaller fragments, once the cleavage is done, are just broken down into even smaller fragments than what they already were, which is why they're unable to be detected regardless of what percentage of polyacrylamide gel he was using. And that's just an interesting fact, not too important there, but... And so after everything, all these experiments, the conclusion that he had is that The bacteriophage, the construction of the head, is far from just taking one protein, attaching it to another one, and so on and so forth, because there are structural proteins that are chemically altered at the stage of assembly, and those that can go to besides just P23 being cleaved into P23 star, P24 being cleaved into P24 star, and IP being cleaved into IP star. And also, I didn't write it here, but P22 was also cleaved, but as I had mentioned earlier, that he wasn't able to actually ever find the P22 star cleaved version, of course. And ultimately, the whole conclusion here is just that the bacteriophage is very complicated. It's a very complicated assembly process.